Hi everyone, welcome to my channel Srinivasa Biology Tutorials. For this section to discuss NCERT Class 12, the molecular basis of inheritance. We going part 4, search for genetic material. Next we search for genetic material. Even through the discovery of the nuclein by Mischer and the proportion, the principles of inheritance by Mendel were almost the same time. So, with the discovery of the nucleus, the Friedrich Mischer, as well as in the Gregor Johann Mendel, that he explained about as in the principles of inheritance in that both same time only. But the time DNA act as a genetic material look along the discovered by and the proven. So after long time they are discovered as in that DNA. Am I right? Yeah. And after in 1926, after by the in 1926, the quest that determined the mechanism of the genetic inheritance had the reached for as in a molecular level. So in the time they get as in more information about that in a DNA, and previous discovered by as in the Mendel, Walter Sutton, Thomas Hunt Morgan, and the numerous other scientists had in a narrowed and such as in the chromosomes are located on the nucleus most of the cells. So here for example you take as any one particular cell, in that cell they are contained as what in some chromosomes as present. Each and every scientist they are thinking like only. And but the question here, what molecule is the actually genetic material had no been there answered. But here which they can, which one they are exactly they act as a genetic material, no one they given as any answer. So the time there the scientist was Frederick Griffith. They are explained about as a transformating principle. He explained about as what transferring principle. In 1928, the Frederick Griffith, the series of experiment he did as a Streptococcus pneumonia bacteria. See, in this 1928, the scientist was Frederick Griffith. He did as in a series of experiments on the Streptococcus pneumonia bacteria responsible for as what? Pneumonia. It is responsible for as what? Pneumonia. At which time it has a miraculous transformation in that bacteria. See that during the course of his experiment, the living organism bacteria had the change as in a physical form. So when he observed as an when that the inject to as in that some genetic information to as in bacteria into as in mice, what happened? The physical changes will be there noticed in that in a different mice. Let us see here the Streptococcus pneumonia, that is causes as what? That is a pneumococcus. That is when the Streptococcus pneumonia, the bacteria grow as in a culture plate. So he took as in a first one culture plate. In that culture plate, he growing as in the pneumonia bacteria. And after some produced as smoothie colonies, while other one to get produced as in a rough colonies. Or so someone are produced as in the smooth colonies, and someone are produced as what? To get produced as in some are in rough colonies. Like these are the rough colonies. Okay, here the smooth colonies is called as an what? It is in S strains. We call as an S strains, and the rough colonies we call as what? That is in R colonies. It is in R strains, or the rough colonies are called as an R strain. S strain and R strain. Let us after this. This is because of S strain bacteria having a mucus protein coat, that is in polysaccharides coat, one, while the R does not have. So here the smooth coat has in what? It is in a mucus coat is present, shiny coat. It is what? Shiny coat or mucus coat means that is in a contain what? It contain as in a poison or toxins. It contains what? Poison or in a toxic. And it is mostly that S strain is in a virulent also. Here once in this injective as an S strain and R strain introduced into as a their mice. The mice infected with as an S strain virulent, what happened? Die from as a pneumonia infection. But the mice infected as an R strain, here it is not developed as any pneumonia. So here in this S strain to get injected as in a mice, mice will be what happen? Get causes pneumonia. But R strain is completely not, they are infected as in a mice. Let us see here. First, in this process, S strain injected as in a mice, mice will be the what happen here? Mice to get die. And R strain to get injected as in a mice, here mice what happen? It is complete, they are alive. Here why? The Griffith here Griffith was able to kill the bacteria by heating them. He observed that in a heat killed S strain bacteria injected as in a mice did not kill them. When he observed, here first he saying about as what? He taken as in a same S strain and he injected as in a mice. Here mice here what happened? Alive. Why? Because when you heated, what happened in this mice? S strain is go to as an inactive condition. It go to as what? Inactive condition. And when he completely, when he they are injected as in a mice, heat coated as well as R strain into bacteria into a mice, mice to get what happened? Die. Here more over the recovered living bacteria from as in a dead mice. So finally he noticed as what? See here once. Here S strain heat killed as well as an R strain. In this both are injected to as mice, what happened? Mice to get die. Why? Because when you observe here, mostly S strain, that is a heat and killed. But here the inactive condition, when they get add with as an active condition, then what happened? Then automatically the S strain mucus coat again they get activated and they can cause this as a pneumonia. Okay, that is next. After when you observe here, the injected as in a mice, the heat killed yes, and the live or bacteria, the mice will be the die. So before only we discussed that one. So the finally the mice will be to get die. And moreover, the he recovered living S strain bacteria, their bacteria will be their dead mice. Okay. Next, finally he concluded that as in a R strain bacteria had as in a somehow way that transformed as in a heat killed to S strain bacteria. Some transformation principle, the transfer of the heat killed S strain had able to the R strain, only synthesis of in a smooth polysaccharide coat become is virulent. So here when here completely is a R strain, it is in a live, and S strain here S strain it is in a when you heat and killed again to get add the R strain, what happened? It is they are completely R strain to S strain. Here some they are transferring and they get finally the mice will be to get die. He noticed. He noticed. Here this must be due to the in a transfer of the genetic material. So here R to S the some genetic information they get pass. R to S some genetic information will be to get pass. However, the biochemical nature of genetic material was not they are defined from his experiments. But here which one they are carrying? Here this R strain to S strain the genetic material are passing. But which one they are carrying, he not get explained. Okay, let us next the biochemical nature of the biochemical characterization of the transforming principle. Biocharacterization of transforming principle. The prior to the work of the Oswald Avery, Colin MacLeod, and their McLean MC Corty in 1933 to 1944, the genetic material was through as in a protein were linked to determine the biochemical nature of the transformation principle in the Griffith experiments. So when they did as in the Griffith experiments, they are rediscovered as in they did as an experiment. The scientist was in there Oswald Avery and MacLeod and MC Corty. In these scientists they are rediscovered as in they are which one they get transferred, which one they are transferred as in R to S. He noticed here. First, they are purified the biochemicals. They are proteins, DNA, RNA, etc. From the inward heated killed S strain seen which ones could be the transfer as in a live R cell into as in a S cell. So R cell to S cell, which one they are transferred, they separate as in their biochemicals, and they discovered as in a DNA alone form as in a S bacteria causes as in a R bacteria to become as in a transfer. So R bacteria to S bacteria, only few are their DNA molecules only they are transferring. And they discovered that as a protein digesting enzyme, that is protease, and RNA digesting enzyme, that is RNase, did not their effect in the transformation. So for example, when you take as in that there are three chemical compositions. Here when you take as in the three chemical compositions, in these three chemical compositions they contain as what? They contain as in a genetic material. Here they contain as in what? The genetic material is in a biochemicals. Okay, in this completely the biochemicals as present in this total biochemicals, that is in a protease, they get added as in a what? In the first they get added as a protease. So when the protease they get add, what happen? The protease enzyme to get digested as any proteins. Here RNA is to get digested. Here RNA is they digested as any RNA only. Here, so the transformation substance is not proteins or RNA. Why? Because here transformation is not going on. But digestion of the DNA did not inhibit the transformation. So here when we are adding with as in the DNA is what happened? The DNA is we get add, here transformation is not inhibiting. And the suggestion as in the DNA causes that in a transformation. So the DNA get act as in a transfer the genetic information from as in a 1 to 1. They conclude as in a DNA that in a hereditary material but is not in a biologist where they convinced. So they complete the final conclusion is what? The DNA get act as a genetic material, get transfer of the genetic material from as in a here to here. But other scientists are not completely not convinced. Okay, this is in a completely biochemical characterization of in a transformation principle. Okay, the genetic material is DNA. The genetic material is DNA. The unequal proof of the DNA is a genetic material came from the in the experiments the Alfred Hershey and Martha Chase in 1952. So the genetic material is DNA. They first did as an experiment. The scientist was in a Hershey and Chase. They worked as within virus. The infected bacteria is called as a bacteriophages. Already we know when that particular bacteria they causes as any viruses. This is called as what? It is a part of what? In a bacteriophage. It is completely called as what? Bacteriophages. Let us here the bacteriophage attached to as in a bacterium, its genetic material then enter to the bacterial cell. So this once you observe here, it is in a virus. Am I right? It is completely what? It is in a virus. It is in a virus. It is a bacterial cell. Here the enter the virus genetic material, they enter into as what? In a bacterial cell. They enter into as in a bacterial cell. Then what happened here? The bacterial cell treat as in that virulent, the viral genetic material. After it was in own the subsequently the manufacturing the more virulent particles. So then finally what happened in this total, their virus means this in a bacteriophage to get developed to get produced as in a number of virulent particles are developed on the in a bacterial cell. Okay, and the Hershey and Chase the work to discover with was a protein, the DNA from as in a virus, they are enter into as in a bacteria. So the when the virus genetic material they are enter into as in a bacterial cell, they can start with as in their multiply virulence is get producing. And mostly in this experiment he did as in a they grow in some viruses in a medium that contained as in a radio phosphorus. In some other medium, they are contained as in a radio sulfur. So he took as in the two mediums. It is in a one phosphorus and the other one is what? Sulfur medium. And the viruses grow as in that presence of the radioactive phosphorus contained as in a radioactive DNA, but not as a radioactive protein, because that contains the phosphorus but protein does not. Here mostly phosphorus here in this medium NH4Cl, it is completely N15 medium, and it is a in this totally in this phosphorus is contained as only for DNA contained as what? Only for the phosphorus, right? Here the DNA contained as only for phosphorus, the sulfur does not only contain as what? It contains as only for any protein. Okay, this saying that one only. Here similarly the virus growing on the only for in a sulfur contained medium, radioactive proteins but not any radioactive DNA, because the DNA does not contain as any sulfur. So sulfur does not contain as any that DNA. Okay, this is simply saying about as any proteins means, sorry, in this part of the phosphorus contain as what? The nitrogen bases, nitrogen base, pentose sugar and phosphate group, and then phosphate group in this present as in where? In the DNA is present. Yes, na DNA of the phosphate group is present but sulfur is not present. Next, here in this radioactive phase, in this the radioactive phase where they allow as an attachment of Escherichia coli bacteria, then is the infection they are processed. The viral codes we are the removed from as a bacteria by as they are irrigating them in a blender. So in this bacteriophages, the virus particles were separated from as in that bacteria, they are spinning them in a centrifuge. So when the bacteria are to get attached here, see first it is in a radioactive, the labeled in a protein capsule, yes, that is in a completely yes proteins, it is in a contain radioactive is in a P medium, means phosphorus medium. So here infection will be going on. First that it is a bacterial cell, it is in a first bacterial cell. In this bacterial cell, it is to get first infected for as what? It is in a completely bacteriophages. It is this process called as what? Infection. After infection, what happened ma? After this enter the genetic material, they enter into as the bacterial cell. They enter into as a bacterial cell. So here what happened? After in this centrifugation process, here the DNA replication is not takes place, but here to get produced as a number of DNA particles to get produced, because here DNA contain what? DNA contain as what? In a their phosphate group is present. So here radioactive not detected in a completely in a supernet. Here the no radioactive detecting in a supernet. Okay, this is in a complete RCHS experiment remaining to discuss in next class. Thank you for watching. Like, share and subscribe.